everyone. Now let us discuss the next topic of biomolecules, the last topic of biomolecules, which is 9.12.4. It is factors affecting enzymatic activity. Factors affecting enzyme activity. Now, enzymes are proteinaceous substances. Enzymes are proteinaceous substances, and these proteinaceous substances, they are active in their tertiary conformations. Now, they have to be in their tertiary conformations to do their maximum activity. So then, what are the factors which can influence the enzymatic activity? Means it goes like the first factor, what is the effect of pH? What is the effect of pH on enzyme activity? Is our first factor. Now, what is pH? pH is a negative logarithm of H plus ion concentration, right? Now, when we take a plot and when we see by plotting the pH scale on the x-axis, this is x-axis, and by taking enzymatic activity on y-axis, or potential energy we can write, or enzymatic activity we can write. We'll write enzyme activity on y-axis. So 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. This is a pH scale range. Now, it goes like, in most of the cases, the enzymes have an optimal pH of neutral. So, what do you mean by optimal pH? The pH where the enzyme shows its maximal activity is called optimal pH. Optimal pH means where the enzyme shows maximal activity. Is called optimal pH. Now, if you have to see optimal pH of some enzymes, take salivary amylase present in the saliva. Salivary amylase is also called ptyalin. It has an optimal pH of 6.8. Then, if we take pepsin, which is present in digestive juice, its optimal pH is 1.8. If you take trypsin and chymotrypsin, its optimal pH is 7.8. So we have listed down few enzymes and their optimal pH. But in most of the cases, the optimal pH, where it shows maximal activity, it will be in the range of neutral pH. Now, so when you are increasing the pH by 1 point, the enzymatic activity also will increase, it will increase, it will increase, it will increase till you get the optimal pH of 7. When once the optimal pH is reached, beyond that also if you are increasing the pH, then the enzyme stability is lost and the enzymatic activity keeps decreasing. So it keeps decreasing. Now here is a place where it has shown its maximal activity. It has shown its maximal activity and that pH range will be neutral range. So this is the optimal pH. Now, it shows what is a graph when we are plotting pH on x-axis and enzymatic activity on y-axis. We are getting a bell-shaped graph. Now, let's work out the second factor. This is the effect of pH on enzymatic activity. Let us see the effect of the second factor, effect of temperature on enzymatic activity. It's almost the same as that of the case of pH. Effect of temperature on enzymatic activity. Now, in the same way, we can tell that majority of the enzymes have an optimal pH within the range of room temperature, that's 35 to 40 degrees temperature. Now, when we do the similar thing, taking temperature, increasing temperature on x-axis and enzymatic activity on y-axis, then there's a thumb rule in biochemistry, which says that for every 10 degrees rise in temperature, the enzymatic activity will double. 10 to 20, again double. 20 to 30, further doubling. 30 to 40, further doubling. Optimal temperature reached. The optimal temperature is 40 degrees. Beyond 40 degrees, even if you raise from 40 to 50, the enzymatic activity becomes half half, half, half. Then it becomes bell shape only. So from 0 to 10, enzymatic activity is this much. 10 to 20, it has doubled this much. 20 to 30, it has doubled this much. 30 to 40, it has doubled this much. Now 40 to 50, I need to go here. 30 is here, 40 is here. Now after 40, if you are increasing the temperature to 50, it becomes half. 50 to 60, it becomes half. 60 to 70, it becomes half. When you join all these things, you will again get a bell-shaped graph. You will again get a bell-shaped graph. That means effect of pH and effect of temperature on the enzymatic activity shows a bell-shaped graph. This is the point where the enzyme has showed its maximal activity. It has showed its maximal activity, and that particular pH is called as, that particular temperature is called as optimal temperature. So most of the enzymes show an optimal temperature around the room temperature. But there are certain organisms, certain bacteria which grow in hot springs, which are called thermoacidophiles. So they have an ability to grow at higher conditions. Now what happens if you increase the temperature? If you increase the temperature, then what happens? By every 10 degrees rise in temperature, you know, enzymatic activity will double. But it's up to a certain extent. It's up to a certain extent. What is that extent? The extent is till the optimal temperature is reached. Now, beyond optimal temperature also, if you are going from 40 to 50, 50 to 60 and 70, beyond optimal temperature, for every 10 degrees rise in temperature, enzymatic activity becomes half. The enzymatic activity becomes half. So this is the effect of temperature on the enzymatic activity. So factors affecting enzymatic activity, we have seen the first factor, effect of pH, second factor, effect of temperature. I talked about certain bacteria which are growing in hot springs. Only for these bacteria, those bacterial enzymes are thermostable. Rest all are thermosensitive only. At higher degrees, at higher temperature, enzymes gets denatured because the tertiary structure is lost. It will become secondary and primary structure. At higher temperatures, what happens at higher temperatures? Enzymes are denatured. What do you mean by denaturation? Bonds are broken. What do you mean by bonds are broken? Tertiary structure becomes secondary structure becomes primary structure. It becomes a linear structure. That active site and all will be lost. Now let's work out the third factor. What is that? Effect of substrate concentration on enzymatic activity. Now this topic is there in page number 157. This topic is there in NCERT, page number 157. It is under the figure 9.7. It's under the figure 9.7. So this pH A, they have given the picture. This they have given B. Let's go with the third picture. What is the effect of substrate concentration on enzymatic activity? Now, for understanding, let us take, we are having 100 enzyme molecules. Now, what is the effect of substrate concentration on enzyme activity? What should we take on x-axis? What should we take on y-axis? Take the velocity of reaction on y-axis. And take the substrate concentration on x-axis. Now we will think that we are going with a situation like we are having 100 enzyme molecules. So we'll increase 10 substrate molecules. So when we keep on increasing the substrates, 10 substrates will react with 10 enzymes. 90 are still there. 20 substrates will react with 20. 50 substrates will react with 50. 75 substrates will react with 75 enzymes. 100 substrates will react with 100 enzymes. So if you see, there is a point where all the 100 enzyme molecules get saturated with the substrate molecules. Now, from 100, if I go to 110, then the enzymes are not free to react with the substrate molecules. Then reaches a point of saturation where the enzymatic activity will be maintained. So this is a point where all it gets maintained. Now this point is called as, what is this point called, where it has reached its maximal activity? And the shape of the graph is called parabola. The shape of the graph is called parabola. Okay. Now, this is the substrate concentration where it has reached its maximal activity. So, this is the substrate concentration where it has reached its maximal activity, 100. Now, 50, there will be a point. 50 means somewhere around here. At 50, whatever the substrate concentration is there, it is called Km. So, what is Km? Km is a substrate concentration not at full maximal velocity, at half the maximal velocity. So what is Km? Km is the substrate concentration at half the maximal velocity. Substrate concentration at half the maximal velocity. What is this Km? Km is a constant. Who derived this constant? Two scientists. Their names are Michaelis and Menten. So they have derived an equation, they have derived a constant, that is Km constant, Michael-Menten constant. The equation, it goes like V is equal to Vmax into S by Km plus S. This is the equation which you call Michael-Menten equation. Kinematics of enzymatic activity. So this is the effect of substrate concentration, which gives parabola. pH and temperature gives bell-shaped graph, right? Now this is the effect of enzymatic activity on enzyme concentration. The last factor which affects enzymatic activity is what is the effect of modulator or inhibitor, other substances, on enzymatic activity. That's our fourth talk. The next one will be, this is the third one. The fourth one will be the effect of other substances, like inhibitors on enzymatic activity. So when it comes to the role of inhibitor, let us talk about two important types of inhibitions. One is called as competitive inhibition. Other one is called as non-competitive inhibition. In competitive inhibition, there is competition between the substrate and the inhibitor to bind to the limiting binding sites of the active site. So here is an enzyme. This is the pocket which is called active site. Now this is an enzyme which is having an active site, and now here is a substrate molecule which can pretty well fit into the active site of an enzyme. And competitive inhibitor, if it should also fit into the active site, it should also have resemblance with the substrate particle. Here we go. So this can also go and fit into the active site. That means in competitive inhibition, the first criteria is there should be structural resemblance between substrate and the competitive inhibitor. There should be resemblance between the substrate and the competitive inhibitor. Now, if the substrate strength is more, what do you mean by strength? Concentration. If the substrate concentration is more, then the substrate goes and binds first. Then results in formation of ES complex. Now, if ES complex is formed, then the substrate is transformed by the enzyme into product. Then the substrate gets converted into of product and enzyme will be released out. So, but if the inhibitor concentration is more, if the inhibitor strength is more, means the inhibitor concentration is more, then what happens? You know, its strength is more, it will go and bind. If inhibitor goes and bind, enzyme inhibitor complex will be formed. If enzyme inhibitor complex is formed, then product is not formed. No formation of the product. Product is not formed, right? So now, but we tell competitive inhibition is reversible. Competitive inhibition is reversible. How can you reverse this enzyme inhibitor complex means? Why did EI complex form? Because its strength is more. Why can't we increase the center of the substrate? We'll do that. So we will increase the substrate molecules, right? If substrate concentration is increased, if the substrate concentration is increased, then EI complex, enzyme inhibitor complex dissociates. EI complex dissociates and ES is formed again. When ES is formed, product is formed. When ES is formed, product is formed. That is why we tell competitive inhibition is reversible. So when we are talking about competitive inhibition, we need to talk about two things. What happens to the Vmax value and what happens to the Km value. So are we reaching the maximal velocity? Somehow, are we making the product by increasing the substrate concentration? Yes, we are doing. That means Vmax we are reaching. That means Vmax, it remains unchanged. Whereas for doing this Vmax, for getting the Vmax, what are we doing? We are increasing the substrate concentration. If you increase the substrate concentration, Km value also will increase, right? So that means we need to tell that the Km increases. Km increases and Vmax remains unchanged in competitive inhibition. It remains unchanged in competitive inhibition. Now, let us take an example for competitive inhibition. So, a very good example for competitive example, the classical example for competitive inhibition is there in the Krebs cycle. It's there in the Krebs cycle. The reaction is conversion of succinate to fumarate by an enzyme called succinate dehydrogenase. By an enzyme called succinate dehydrogenase. Succinic acid is a 4-carbon compound having 2 carboxylic groups. Now, succinic acid is a 4-carbon compound having 2 carboxylic groups, which is the substrate molecule. This succinic acid is the substrate molecule. We told that in competitive inhibition, the inhibitor should resemble, right? So, let us take the name of an inhibitor which resembles. Malonate. This point appeared in 2017 NEET examination. So the inhibitor is malonate, and malonate also is a 4-carbon compound having 2 carboxylic groups, means the functional groups are same here and here. So malonate also competes with to bind to succinate dehydrogenase. Succinate also competes to bind to this. If succinate binds, fumarate is formed. If malonate binds, fumarate is not formed. So who is the substrate? Who is the product? And who is the inhibitor, they will ask. So the fumarate is product. This is one classical example for competitive inhibition. Another example is in order to control the bacterial infections in our body, the antibiotics what we take act as competitive inhibitors to block the bacterial metabolism. So to control bacterial infections, the medicines act as competitive inhibitors to the medicines act as competitive inhibitors to bacterial reactions or bacterial enzymes. This is about competitive inhibition. Now let us talk about non-competitive inhibition and then we can end the session. Competitive inhibition, we told that there is resemblance between the substrate and the inhibitor, and either ES complex will be formed or EI complex will be formed. But in non-competitive inhibition, so it has 2 sites. The enzyme has 2 sites. One site is an active site. The other site is an allosteric site. Now let us talk purely about non-competitive inhibition. In non-competitive inhibition, the substrate and inhibitor do not resemble each other. They do not resemble each other. And the substrate binds to active site of enzyme, while the inhibitor binds to another site, which we can call it as allosteric site of enzyme. It binds to allosteric site of enzyme. If we see that this is an enzyme. Now this site is allosteric site. This site is active site. To the allosteric site, inhibitor will bind. To the active site, substrate will bind. Both are binding to their own places. So, ESI complex can be formed. Now, when the inhibitor binds to the allosteric site, it makes some changes in the substrate so that what happens, the substrate can no longer bind to the active site, the substrate drops down. The substrate drops down. When the substrate drops down, no product formation. When the substrate drops down, no product formation. So this is about non-competitive inhibition. Now when we have to talk that in non-competitive, it is reversible or irreversible. First difference, it is irreversible. Non-competitive inhibition is reversible. What happens to Vmax and what happens to Km? Are we reaching the product finally? No product. Means Vmax decreases. And are we increasing the substrate concentration? Is it so because substrate strength is low, it's falling down? No, not like that. So that means by increasing or decreasing substrate concentration, we cannot alter the rate of reaction. So that's why Km remains unchanged. Km remains unchanged and the Vmax decreases. And it is a irreversible reaction. Means you cannot bring back the reaction to a normal state again. So this is about the topic 9.12.4 factors affecting enzymatic activity. In the next class, we will talk about the nomenclature and classification of enzymes and cofactors. Hope you like the lecture. If you like the lecture, you can like, share and subscribe to my channel. Thank you.